I'm Patricia Kabitzke, Senior Scientific Program Manager at Cohen Veterans Bioscience, and I'm involved with driving our preclinical program forward. Cohen Veterans Bioscience has a very specific interest in developing translational tools that will de-risk drug and therapeutic development and help us understand the biology of disease. We know that preclinical animal model systems are central to accomplishing these objectives. In recent years, however, we've become increasingly aware that results observed in preclinical studies often fail to generalize to the clinical setting. These late-stage failures are due in part to the lack of construct and predictability of preclinical animal studies. Thus, the purpose of today's panel is to discuss some of the issues associated with PTSD translational models. Let me start by introducing our first panelist, Dr. Lois Winsky. She is the Chief of Molecular, Cellular, and Genomic Neuroscience Research Branch with the Division of Neuroscience and Basic Behavioral Science of the National Institute of Mental Health, where she oversees also the psychopharmacology program. Dr. Winsky, I think you might agree that current animal model systems of PTSD appear to have significant limitations, including weak validation. What do you think about the state of translational models today, and what efforts have you been pursuing to develop a next generation of models?
Thank you for the question. I appreciate the opportunity to participate today. We do know that we need model systems. They're an essential component of a therapeutic development program. They're required for safety studies for advancing new therapeutics into the clinic. They're also very useful in preclinical therapeutics to identify evidence, to provide evidence of target engagement, as well as to give us initial ideas of pharmacodynamic properties and to help us set dose in the first studies in people. We also know that the preclinical systems are very important for understanding the underlying biology, and a lot of that interesting science has really been identified in the last 2 days. And in that sense, because we know about the circuits that underlie, at least we think we know about the circuits that underlie components of our deficits, then we can identify novel targets in preclinical systems. But historically, we know that it's very risky to try to predict effects in people based on preclinical measures, particularly in the area of efficacy. And that really matters, as you would remember maybe from Magali's slide earlier today, that those preclinical measures are what really set the pipeline and determine what goes forward and what doesn't. So I have an advantage today because a lot of what I'm going to say, or at least a lot of it, will really resonate with what's been discussed the last 2 days. The NIMH is taking some various measures, partly things that were mentioned by Holly Lisenby in her talk this morning, but really moving towards understanding what is it that contributes to the pathophysiology for lots of disorders and the functional deficits. The goal is to move towards a precision medicine model where we understand what the basis of the heterogeneity is, but also to understand what are the factors that are really critical for determining the loss of function in daily life. So you also heard yesterday and today about the RDoC effort. So the RDoC effort is really to understand functional domains and to link those with biology. And so so the idea then for the experimental medicine approach is to really then target those systems that are defective and that matter. And you can use preclinical species for that part, but then also when you go to test the new therapeutics in people, the experimental medicine approach aims to identify, to give evidence of target engagement, but also to identify individuals who might be more likely to respond, understanding that it's possible we could have a therapeutic that would only work in a subset of people. No, I need to go back on that. So, the animal measures that we have in place are really not tied to the biology. The problem is that it's actually two problems. One is that we don't understand the biology of the disorder, which has been made very clear in the last 2 days. But the other part is that the measures that we use in preclinical species, for the most part, don't align with the biology that we do know. So what we're trying to do in the NIMH is develop a program to get a good idea of where we may have coherence of measures and where we may know if there is conservation of circuit function related to deficit areas in the context of biology. So the NIMH has a program to develop novel assays to address translational gaps in the therapeutic development pipeline. And really, the goal of this is to look for measures of neurobiological mechanisms like EEG that we've already heard about, and to see if we can optimize those measures in both healthy humans and healthy animals that are used in a preclinical development pipeline. And then the second component is to evaluate the performance of those assays in response to more than one level of apertibin. And so the idea of this is to really try to build some assays that may predict across that gap that we've already heard about whether a preclinical study is going to predict an effect in people or not. And the other important point of this is that the data from these projects will be put in the public domain regardless of outcome, and that's really important because that way, if there's an assay measure that doesn't work, then we don't have a repeat of people keeping to try to do it over and over again. So this is just an example, and actually it's kind of perhaps relevant to some components that we've heard this afternoon or this morning. And it's an example of the type of neurophysiological measures that we're thinking of targeting. And this is just showing you a sleep spindle. A sleep spindle is really a landmark measure of non-REM sleep. And it's something that we can easily measure in both normal humans with a scalp EEG and with a depth electrode in rodents. And it's highly conserved. And it actually changes in learning and memory, and it changes in age, and it changes in certain kinds of cognitive pathology. But it's really meant just to give you an idea of the types of measures that we're really trying to evolve.
Excellent.
Thank you.
Thank you so much.
Yeah.
Our second panelist is Dr. Lindsey Grandison. He is on the faculty in the Department of Physiology and Biophysics at the Robert Wood Johnson Medical School and is a program officer at the National Institutes of Health, National Institute of Alcohol Abuse and Alcoholism. Dr. Grandison, can you tell us about how you were thinking about the problems of comorbidities associated with PTSD, such as alcohol abuse, and how they might be addressed in animal modeling systems?
NIAAA has looked at this as a priority because it's a real gap issue. There's a high coincidence of PTSD and substance and alcohol abuse, and it's because of the high incidence and because of the fact that it really exacerbates the symptom severity and makes treatment much more difficult. It's a problem, and it really hasn't been examined because one of the most predictable exclusion criterion for clinical studies of PTSD is alcohol use. So there's been very little work done clinically to look at the comorbidity, and there is very little work that currently exists on the comorbidity in animal studies because partially the animal models which have been used in PTSD have not been really validated for alcohol studies. So we're very interested in trying to develop a valid animal model or partitions of that symptom that could be used to study the relationship to the comorbid condition. In addition, there's so much overlap between the consequences of PTSD and alcohol dependence. Some of the genes that are risk for PTSD and for substance abuse, alcohol dependence are the same. A lot of the perturbations in biological processes are the same. The changes in the amygdala and the lack of connectivity or decreased connectivity with the prefrontal cortex are similar. So what we would like to do is use this comorbid condition to look at the processes that are taking place in this to identify how PTSD is increasing the risk or the development and progression to substance use, alcohol abuse, see if there's any way to intervene and prevent that progression, see what those mechanisms are. So we see it as a fruitful area that's of significance, and that really is a gap in our research efforts at this point.
Certainly, I think many in the audience would agree. Dr. Kerry Ressler certainly doesn't need much of an introduction in this room, but he's our third panelist. He is the professor of psychiatry at the Harvard Medical School and the James and Patricia Poitras Chair in Psychiatry, Chief Scientific Officer, and Chief of the Division of Depression and Anxiety Disorders at McLean Hospital. Dr. Ressler, as co-chair of the new AMP It Up initiative with Cohen Veterans Bioscience, can you tell us a little bit about what this initiative is about?
Sure. Thanks so much for having me, Patricia. And first, just thanks to the Cohen family for supporting this amazing 2 days, and Magali for all of your work over years in this transformative effort. I think it's really going to change the world, so thank you guys. I'm delighted on behalf of myself, Israel Lieberzahn, and about at least 20 other really researchers around the country, and we probably have at least one international person, so we can say around the world, to talk a little bit about AMP It Up. So this is a cute and very difficult to remember the acronym's easy, AMP It Up. What it stands for: Alliance for Models of PTSD, Innovative Technologies and Uniform Practices. Hopefully everyone got the, the USB charger, so thank you guys for the AMP It Up reminder there. So what is this about? So it's really a open, translationally relevant, open source group that's trying to figure out how can we pull all of these basic, really leaders in basic science of models of stress, fear, and PTSD together to transform where we stand in the field for translation. I've been talking with folks as we Amit showed the 600 and something thousand ways to get to PTSD, right? And I wonder if we should take a lead from the autism spectrum disorders and change our acronym to post-traumatic spectrum disorders, you know, to give it to, as we need to keep in mind that there is not one thing we're modeling, but there are many things. So I think the first take-home point of the people who study these disorders is that we're not going to make an animal or a mouse that has the same PTSD that a human does. There's just way too many things going on. But what I think we have going for us in the area of PTSD basic research that I think is far exceeding many of their other areas of psychiatry is many shared components. We'll walk through the amygdala and some of the shared known emotional circuitry related to fear and trauma and stress. We know when it starts. We don't know in any other psychiatric disorder, we don't have the same level of control for studying the longitudinal progression as we do with when it starts. And we have over 100 years of basic science research and behavioral research, starting with Pavlov, in understanding these processes. So our goal is really to develop models in a broad variety of terms. We've done this through several ways, led by Patricia and Magali. We started with a preclinical workshop in the spring. And the goal there was to address the challenges of PTSD translational science in order to establish this preclinical roadmap to advance new therapeutics. When we think about what are the constructs we can model, there are many, and many of these have quite a lot of overlap between animals and humans. So I'll talk some more about fear memory momentarily. We heard from Israel about context regulation, obviously the sleep and circadian rhythm processes, and Dr. Winsky showed some of the sleep examples a minute ago. And then once with Amit gave a beautiful presentation on circuit function, as did Israel. And again, the circuits that, even though obviously a mouse cortex is not a human cortex, much of the same connectivity is there across the limbic system, across the basic emotional and fear processing. So we can have models that are primarily focused on circuits, primarily focused on cells, and primarily focused on some of the behavioral constructs that are dysregulated with trauma. One example, again, how to think about this is again, that there is not going to be one model, but there are going to be many models. And how do we find the points of intersection for these models that will help us identify what is best for translation and what may help with precision medicine explain some set of symptoms in some people and maybe a different set in other people, and how those then biologically are intersecting. And Pavlovian fear conditioning is an example that we can measure fear using processes like fear-potentiated startle that we can measure in a mouse and a rat by decades of work by people like Mike Davis. And we can do very similar fear-potentiated startle in a human. And we can even do very similar parallel fear conditioning, expression of fear, extinction of fear. And lots of data now show that fear is dysregulated in people with PTSD. If you take someone with PTSD and you put them in a laboratory setting, they'll show an enhanced learning of the fear process, overexpression of fear often, and often a deficit in extinction or recovery from fear. And so those are all laboratory biomarkers, if you will, of the process. Again, one area that a lot has been based on and that we understand a lot about is the amygdala. Israel yesterday showed us the prefrontal cortical, cingulate, hippocampal, and amygdala circuits that may regulate different aspects of symptoms. And what's exciting, and has been exciting to me for 20 years since I started working in this area about the amygdala, is that the known hardwired circuitry to the subcortical areas from the output regions of the amygdala create a fear and panic attack that that we can model and that help us really understand what this hardwired circuitry is. So that's one example of how using classic fear conditioning, using classic models in which this circuitry is very well understood and is similar across animals and humans can help us to better understand how this is dysregulated. And likely it's going to be dysregulated in different ways in different patients with PTSD, and likely different gene pathways that are associated with PTSD risk are going to be broken, if you will, in different aspects of these pathways. And at the next level, beyond circuits, what animal models give us the ability to do now is start to understand the very precise microcircuitry. We used to say the amygdala, and now we know that the amygdala, I still say the amygdala, but that it's really a cluster of about 10 or 15 different subregions. And each of those regions is made up of many different subtypes of cells as well. And we're to the point in both human models, and you'll hear about the brain bank from Sabina shortly, both in animal models as well as soon in humans, be able to do cell type specific profiling of different molecular pathways. And it may be that that's really the level of specificity we need to really start understanding how the genetic and biological risks for PTSD occur. So again, the Amp It Up group began with the first preclinical workshop in February. We've had a number of very productive and exciting phone calls. Thank you again, Patricia, for your great organization around all of this. And have another preclinical workshop being planned for October that will be near here, within the goal of potentially announcing an RFA for specific approaches to helping with the preclinical pipeline and preclinical roadmap going forward. Thank you very much.
Thank you so much, Kerry. Our fourth panelist, Dr. Emer Leahy, is CEO of Psychogenics Incorporated, a profitable preclinical CNS service company. Also CEO of PGI Drug Discovery, LLC, a company engaged in psychiatric drug discovery with 3 partnered phase 2 clinical programs, and adjunct associate professor of neuroscience at Mount Sinai School of Medicine. Dr. Leahy, you lead a contract research organization that works on standardization of these model systems, and you work with the pharmaceutical industry. Can you tell us a little bit about your practices, in particular, how you focus on standardization?
Absolutely. So standardization is incredibly important in our business. The company has been around for the past 17 years, and I think that's important because it speaks to quality, and that speaks to standardization. We provide a service to pharma, biotech foundations. We have relationships with over 140 companies worldwide, and about 90% of our business comes from repeat clients. And it all stems from how we conduct our research. So let me just highlight some of the important aspects of standardization, all with an attempt to reduce false positives. So, unfortunately, I can't read this, but we certainly have to pay attention to every aspect of what we do in the lab. Everything from husbandry, how the animals are housed, the environment that they live in, how we conduct our studies, make sure that they're randomized, that the investigators are blinded to the treatment groups and to any aspects that we can control. And make sure that we always include positive controls where possible. That could be a positive reference compound, or it could be another type of treatment, or another type of lesion or model that we're including in our studies. We try to QC every aspect of what we do. So it's everything from when the compounds come in, they get labeled, they get barcoded, the drug prep gets QC'd, the study gets QC'd, and then of course on the data collection and data analysis side, very important. SOPs, we're very transparent as a company. We invite all of our clients to see what we do. They can review our SOPs, our training records. We retrain staff on a regular basis. We also, when we work with foundations, and I've included just one example here from CHDI, we will include SOPs on their websites as well and provide them with all the details of the work that we do. And here you see in the middle of this slide, it's a working guide to Huntington's disease, how we handle animals specifically for Huntington's disease, and it goes into all of those aspects that I just mentioned, the husbandry, the environmental conditions, the testing, etc. On the testing front, when we look at an animal model, we try to look at the model as broadly as possible. So we don't just hone in on a single endpoint measure. So for behavior, for example, we try to examine the behavior as globally as possible. And we've developed some platforms that enable us to do this, and we complement those platforms with more standard behavioral tests. So looking at anxiety and cognition, fear response, those sort of things. We complement behavior with things like EEG measures to tap into the circuitry, microdialysis, immunohistochemistry, and then obviously to imaging as well. And trying to understand how circuitry relates to behavior, very important. Reproducibility is always a challenge. When we offer a client a particular disease model or a test, we've always run that model multiple times in our hands, and we consider it reproducible. We also include reference standards, and in the event that a reference fails to produce the desired effect, we will repeat that study at our expense because, again, we're a service organization, so you live or die by the quality of the work that you produce. Automation, very, very important, and what we've done, at least on the behavioral front, is to automate behavioral capture and analysis. And in doing so, we've been able to not only standardize how we do the work, but eliminate bias and also improve statistical power. Statistics are another aspect I wanted to touch on. We try to do power analyses in the work that we do so that we know that our studies are fully powered, and we'll always look at multiple endpoint measures, but we try upfront to define the primary endpoint and also define how we deal with outliers or missing data. So putting all of that up front. And lastly, information systems, very important in terms of tracking and making sure that everything is captured. Everything from a compound coming into our lab gets barcoded. The animals, their cage cards are produced, they're chipped. We can track the animals, the compounds throughout. The data gets captured automatically, and then of course we can do the analysis as well online. So the information systems are very important. Just to touch briefly on the automated technologies that I mentioned, we have 3 platforms that we developed in-house. They all combine robotics, computer vision, and bioinformatics. This is important because, as I said, they standardize what the animal goes through in each of these platforms, and the data is automatically captured using computer vision and analyzed with bioinformatic tools that we've developed, all of which then eliminate subjectivity and human error. So SmartCube is a platform that we developed a number of years ago, and I really should give acknowledgement to Danny Brunner if she's here. Danny spearheaded the work in all of these systems, but in particular in SmartCube. It's a system that challenges an animal, and we capture everything the animal is doing with multiple cameras, giving us a 3-dimensional picture of what the animal is doing. And from there, we collect a massive amount of data, giving us that statistical power that I was mentioning. And This is just some data from SmartCube with 2 PTSD models that we've run. The first is a chronic social defeat model, and it's compared to an immobilization stress model. And I think this speaks to a number of points. First, you see that there's a lot of similarity in the data here. So just to orient you, the midpoint is these are behavioral features, and the midpoint is the control value. So anything that's shifted to the left is a decrease in that feature, anything to the right is an increase in a feature. And you can see immediately that there's a lot of features here that are decreased at the top. A lot of these are motor measures and gait measures. There's a gait challenge in SmartCube. And then toward the bottom, you see some differences. These are more anxiety-like measures. There are some consistencies, and then there are some differences between the 2 models. And I think it speaks certainly to the heterogeneity as well of PTSD.
Thank you very much. Dr. Winsky, I'm going to come back to you. PTSD occurs at least as frequently in women as in men. However, most preclinical testing occurs in male animals. Can you comment about what this means for the science that we take forward?
Well, in the past, this has been a real problem because compounds would be tested preclinically just in males and then be evaluated clinically in both species, and often you didn't even know if the drug would have a different profile across sexes. And now, since January of this year, NIH has changed the rules and required that sex be considered as a biological factor in all grant applications to NIH. And that's part of the broader issue of dealing with rigor and reproducibility. It's a really great step forward because we really do need to understand where sex matters and then move on from there.
Does the NIH fund studies that are just powered for male rodents or just for female rodents?
For the most part, not anymore. There are cases certainly where there's a disorder that's maybe just seen in males, but even if it's a genetic disorder that's just in males, often including both sexes, can give you some validation as to whether you're looking at something that's relevant or not. So it's important.
Thank you so much. So some may argue that animal model systems are not similar enough to the human condition and are unlikely to mirror the full extent of PTSD or its comorbidities. Dr. Grandison, how would you suggest we deal with alcohol and substance abuse and other comorbidities in our model systems?
I don't have a real answer. We're hoping that the collaboration amongst all the groups here will be able to provide a better approach. Dealing with trauma and alcohol consumption has proven in the past to be a difficult issue because it's well recognized clinically that there's an association between stress, trauma, and increased alcohol use disorders. If you try to then turn that back into animal models, the alcohol literature is very mixed. There's conditions where people have reported stimulation, decreases, or no change at all. And it's only now that we're really able to develop some rigorous models where they're reproducible, where we get consistent results. So given, plus the fact that alcohol dependency and alcohol use disorders is a heterogeneous condition as well. You have different patterns, people who can drink high levels chronically, people who drink high levels intermittently and binge drinking. And these produce some kind of different patterns of response, different biological processes. So when you look at that and try to do a comorbid study with PTSD, which is itself heterogeneous, it creates tremendous difficulty. There are some animal models of PTSD. And the real issue is trying to relate the kind of studies you will do that have correlations to clinical situations. And I think that's the approach that we would like to follow is pick out not a single model or a single approach, but to use multiple approaches that we can then relate to clinical studies, to the phenotyping studies that others are going to be doing, and try to develop those in parallel with human studies. And then use those to identify pathways that are affected by PTSD that maybe will promote alcohol abuse. And then from that, try to develop interventions, behavioral or medication.
Great, thank you. So Dr. Ressler, as you know, we anticipate seeing the first genome-wide significant risk alleles for PTSD within the next 6 months. How do you see those being useful in developing new animal model systems?
Thanks. So I think there's a number of really exciting approaches that one can take in combining human genetics and really human biology first approaches, whether it's genetics, whether it's other omics, as it were, or circuit approaches. Kirsten yesterday gave a great example of the reverse translation approach with the schizophrenia hit that was in an MHC locus associated with inflammation that then through animal models they were able to show is specifically involved in the complement pathway that's involved in synaptic pruning. And that would then lead to a whole new set of therapeutics that would have never been thought about before if you didn't understand that biology. So I think the first round will be, what are the genes that statistically in large-scale studies are associated with PTSD, and then taking the animal models, that back into the animal models to say, how are these genes being affected? Another way to, that we can think about it is really a gene-first approach to intermediate phenotypes. So, the problem that we're all struggling with is there are many different PTSDs, if you will, but it may be that this set of genes leads to more of, say, a hyperarousal or a sleep dysregulation, another set of genes more to a, cardiovascular effects or whatever. And so that may also help us to differentially define subtypes. And I think another approach is convergence. So basically, can we take the genes or the RNA or the epigenetic signatures in the human studies and then look at the RNA and epigenetic and gene pathway approaches in the animal model studies, both at the whole animal level, but as well in regional level or even specific cell types. And that will, I think, also give us much better tools for prioritizing which pathways are most tractable and most directly relevant in humans.
Great. I'm going to ask you the same question, Dr. Winsky, see if you... And add to it. Sure, of course, of course. So it has to do with the GWAS results that we'll be seeing in the next 6 months. What opportunities do you think might be gained from those results in the preclinical animal models?
Well, the first thing is to be sure that we understand the biology of those molecules that are identified. And we do have special funding announcements to really help us expand that biology, particularly for things that are unknown genes, right? We don't know what the neurobiology function is, and it's very important to put them in the context of what they might do in a normal system before we can expand into understanding the sort of pathophysiology.
Excellent, thank you. Anyone else have anything they wanted to add? Okay, excellent. All right, I think we'll open up to some questions.
Marty.
Great. I think we've got a mic coming to you. Oh, sorry. Up in front. Sure. Thank you.
Thank you. One interesting thing we've observed is using the same insult on the animal, but with three different strains of mice, and again seeing how very different each of those strains react. That is, we chose these particular strains of mice, one of them, of course, because it had been used routinely, but the other two was just what the prion people had done, and we said, well, let's see what we see. We saw rather dramatic differences in terms of these animals reacting to the same exact insults. And I think that is probably something that we'll all have to...
And sometimes it can be the same strain of mice, but from two different sources.
Mm-hmm.
Oh, yeah, right.
Yeah, I comment on that.
But I do think that having different strains where you're actually seeing such dramatic differences give us the opportunity to begin looking at perhaps resilient or a less susceptible type of physiology.
Emer?
Yeah, very important point that you raise, and it's something that we grapple with in everything that we do. We've studied more than 10 different strains of mice in many of the psychiatric endpoint measures that we examine, anxiety, schizophrenia, and other measures, and we see dramatic differences, and it informs us to decide on what strain to use in a particular test or in a challenge. And in PTSD, we've done some of this work as well, and we see very different responses in different strains.
Yeah.
Oh, another question.
The black 6 is used exclusively to study alcohol because they're the only strain that really reduces consumes enough alcohol to reach intoxication levels. And other people have used this approach to develop recombinant inbred strains of black 6 versus DB2, where they're trying to phenotype each one individually and look at how the genetics correlate with phenotypes. So strains are tremendously significant.
Yeah, the other point I would like to add is that if you have large strain differences in your measure, then it could very well be that that's not a translational measure. You know,
Yeah.
just a thought.
I think this is a question for Kerry, but it's for the whole panel. And the prior conversation actually is a nice segue, because I'm wondering how the changes in DSM-5 from conceptualizing PTSD no longer as a fear-based disorder affect animal research. For example, I don't think you can use cats to study guilt or shame, because they can't feel it. So, but at a more serious level, it strikes me that we have a very different way of conceptualizing PTSD, and I actually wonder what model you would use, or models you would use, to capture the broader range of phenotypes that we now have in PTSD.
Yeah, great question. A lighthearted response first, which is that my dog does feel guilt and shame.
Dogs are different. Dogs feel guilt, but cats don't.
And in one more cat question, we saw a number of bioinformaticists talk about the hairball problem, and I think it's an interesting cat. We can model that in cats as well. But I think where much of the sort of basic science is going again is that although we have to, for clinical purposes and to some extent for charging purposes, have a diagnosis, right? And yet we very clearly know that PTSD is not one thing. And PTSD is probably many different syndromes that we cluster together as PTSD. I don't think we're going to have good models for guilt and shame anytime, probably in my lifetime. But I do think we can have very good models for some aspects of PTSD. And I think certainly the fear-related aspects, the hyperarousal aspects, maybe some of the emotional dysregulation, that some of those in the circuit parts, I think we can. And so maybe it will come up that you eventually are seeing somebody for PTSD subtype, hyperarousal, and nightmare subtype, and that we have specific drugs or targets for those subtypes that have been developed through these models. Is it going to get all of them? No, but I think it is maybe a pathway forward where we can start to see progress. Other thoughts?
The only other thought is that, so in the animal models, what they're relying on are physiological endpoints, and the human diagnostics are relying on a lot of behavioral endpoints. So if they can make correlations between the physiologies in the two groups, they're making assumptions that it's producing the same kind of...
Yeah, I think the Colonel, who was sitting there yesterday, made the great point that so much of our translational problems is that we're not translating it to the humans properly because we've been stuck sort of in our self-report measures, and we've got to use all of these other approaches to have better digital phenotyping, intermediate phenotyping of our humans to better understand what we're translating to.
And also understand that sometimes behavior doesn't map onto behavior across species.
Over there.
One question. Yeah. Are there any simple HPA axis markers that you see consistently in animal models that are translated to human models, whether it's, no, I guess the answer is no, huh?
Well, there's probably differences of opinions. I mean, there's the ones that are, nothing's 100% consistent across anything.
Right.
There's certainly HPA dysregulation that you can see in stress models. FKBP5, I think, as more proximal to glucocorticoid regulation has been more robust, I think, than just court levels. And I think some of the CRH has held up in some ways, but again, it hasn't yet translated to meaningful treatments.
Right. Yep, thank you.
Yeah.
Hi, this is a question for Emer. I wonder if you can comment on combining more than different strains, but different transgenic models to start a TBI, so in the hyperphosphorylated tau, for example.
Right, so we have a number of transgenic models that we have in-house, and I guess the question is, can we show an even more robust phenotype by combining some of those transgenic models together to, you know, express symptoms more robustly, perhaps. But specifically with tau, we have a TG4510 model, which is a repressible model. You can administer doxycycline and repress the expression of human tau. And it works beautifully to rescue the phenotype. And I guess the question you're asking, Danny, is can we use that as a model of TBI, where we would induce TBI specifically in that model, and perhaps we get exactly a more sensitive output. And I think that's an excellent suggestion and something that we could certainly pursue.
Hi, Stu Hoffman from VA. I was wondering if any of you are incorporating any brain injury in your models of PTSD to see, you know, like what was brought up yesterday in the talk, that you study extinction or more a greater propensity to develop fear-like or PTSD-like behaviors.
I can comment on that as a starting point. Thanks, Stu. We're going to be collaborating with Ross Safanti at who's part of Partners Spalding Rehab. They have a nice closed head injury model based on a group at Tennessee, Anton Reiner and Scott Helt. Using a similar closed head injury model, they were able to show nicely that prior trauma then leads to a deficit in future extinction of fear, so that the animals have more difficulty with reversal learning as well as extinction learning. And one intriguing tidbit is that it looks like that animal TBI model, that they have a specific dropout of neurons in the amygdala that we think inhibit fear or enhance extinction. And so if that holds up, that would be one sort of combined cellular and TBI model of how TBI might interact with trauma to increase risk for PTSD.
Thank you.
I actually have a question. So many of the current model systems apply relatively short-term stress to normal rodents, which we know is very different from PTSD, where there's likely an underlying genetic vulnerability, and this combines with random and also long-term environmental stressors to produce long-lasting behavioral pathology. So what do we see as an alternative to this approach?
Well, go ahead.
No, please, go ahead.
Well, one area that we have developed is actually alternative model systems, including cell-based systems. We heard a little bit yesterday about iPS cell systems, which are useful, particularly for understanding some genetic risk potentially, although we're just really starting to try to elucidate that. There are also some higher throughput assays that could be used to really understand, if you find a molecule that's associated with PTSD, how that affects neuronal activity and synaptic plasticity and those kinds of mechanisms. So, and even in the gene level, there's potential to use simple model organisms to really screen through multiple gene risk genes to really help you prioritize your research agenda in those areas.
And we're increasingly, Emer showed the immobilization stress compared to this chronic social defeat, sharing some and having some different components. And many, including Steve Marin's group, and we are also, and Israel, of course, are using a more severe stress model followed by a sort of an incubation period, sort of like what Dr. Kandel showed yesterday. And then in those animals, you might be using fear or fear extinction more as a probe of how the brain is responding to that fear and not necessarily as the model of PTSD itself.
In terms of alternatives to animal models as well, I think I've been fascinated with some of the work that Hugo Geertz and others have done in silico. Mm-hmm. It's really quite interesting to see how you can model not just the networks, but the pathways and the molecular pathways in particular.
Yes.
And the other point I'd want to make is that sometimes humans are the best models. So sometimes studies in normal humans can really advance biology understanding. So.
Does anyone have any? Yes.
So I was wondering if you had any thoughts or data about the environment of these animals in terms of enrichment of the environment, or how that contextual, you know, because we sort of discussed that vis-a-vis humans at some point in the meeting, and it just seems to me that, you know, for example, with cognitive enhancers or various kinds of things, you sort of consider the enrichment of the environment or the complexity of the environment, et cetera. So I just wonder if that's been kind of bantered around or at all.
Yeah, enrichment has a dramatic effect on animal behavior. In fact, in Huntington's disease, we enrich the environment. The animals have little igloos and bones to chew on, and they're relatively happy. And we have extended their lifespan by weeks compared to other labs as a result, I believe, of the enriched environment in which they live. Of course, you have to be careful about what you're studying, and whether, you know, if you're studying cognition, for example, you may not want to enrich your environment because you may have a difficult time seeing the deficit that you're looking for. So we do both, depending on the type of work that we're doing.
I think with just our last couple of moments, I wanted to ask each of you what you think is the next most important thing that we need to do in translational modeling to advance the field.
So I would say the next most important thing to do is to understand the pathophysiology of the disorder, the heterogeneity, and to target functional deficits for developing preclinical measures, and then to determine the degree to which there's evolutionary conservation of function of those measures.
Homology, thank you.
I would think validation of the animal models and how they relate to clinical studies, and try to cross-validate and transfer that information back and forth.
Thank you.
I think I would pick up on Lindsey's point, and that I think historically it's very hard at NIH to get funding that for a study that includes both animals and humans. You know, there may be good programmatic need, but you can never get it through study section. And I think processes like the CVB, you know, where we can really in parallel iterate what are the human intermediate phenotypes and what are the animal intermediate phenotypes we can model to really find that sweet spot, I think.
Did you want to respond to that, Lois? No, no, okay.
That's fine.
Emer.
We heard a lot today about mapping circuitry in the human brain, and I think it's fantastic that we're seeing that. We have fabulous tools in animals, of course, and so being able to map circuitry to specific behavioral outputs is, I think, very important, and will help ultimately with the improved translation.